Yes, thank you. I just check my screen now.
It's
So
fine.
I hope it's okay for you.
Yep.
Thank you. So just I take the point. So here. So thank you, Michael, for the introduction. So my name is Magalie Weber, and so my background is more on food sciences and technology, but as Michael told, I am now currently dealing with knowledge engineering. And more precisely, I am involved in ontology building and data stewardship for open science purposes. And this talk was prepared under the supervision of Liliana, who is with us. And Liliana pioneered this work by developing the ontological core model PO2 that we will present in a few minutes. And Julien Cuffy is also with Stéphane Dervaux is also with us, and both are software engineers and engaged on the project several years with expertise in semantic web technologies. So today I am presenting how the PO2 TransformON and FermentON domain ontologies can help us achieve interoperability and reuse of data on bioprocesses. And these are projects funded by INRAE Research Division Transform and also MICA, which is dealing with microbiology, and also supported by the Research Division MATNUM for numeric digital sciences, and with the CATI DISICO group, which is an IT group for knowledge engineering and IT services. So first, I have to mention that numerous collaborators coming from 6 INRAE research divisions and 14 joint research units have contributed to the work. And indeed, the solution we are presenting today is the outcome of a fruitful joint effort involving many different scientific experts in the field of food and bioproducts, as well as experts in data science and software development over the last 5 to 10 years. So first, I am presenting the challenges and solutions, and let's have a tour on PO2 TransformON and FermentON. So the scope of the ontology that we have developed covers all aspects dealing with food, bioproducts, and waste engineering in the context of bioeconomy and global agri-food systems. So as you may know, agri-food systems are complex systems defined as the elements and activities related to the production and consumption of food and their effects and also, of course, processing and marketing. And this also includes many socio-economic drivers, environmental and nutrition and health aspects. And we are on our way towards a circular and sustainable agri-food system. And we are dealing with transformation of bioresources. So in this context, our purposes are to design tasty and healthy food with eco-friendly and sustainable processes, and to design functional bioproducts and exploit waste or residues produced by human activities. So now, knowledge discovery in food and bioproduct engineering is challenging because there are complex research questions on both process performances and product usages. And the data collected are by nature multidimensional as they cover different disciplines, and it is necessary to integrate and exploit large collections of experimental data sets coming from various projects and having heterogeneous sources and formats. And moreover, these data are sometimes incomplete and imprecise. And for the modeling, we have to reason under uncertainty. So how to reason under uncertainty? There is thus a need for structuring data and providing information about the context in which the data was obtained. And so we have to standardize the metadata to facilitate their discoverability and reusability, to query and assemble the data into new data sets, to share and reuse them. And for these purposes, the FAIR principles and the semantic web technologies are offering solutions for data which is findable, accessible, interoperable, and reusable, as you may know. And this enables finally statistical modeling and machine learning for knowledge discovery. So Data interoperability enables machine learning engineers to train multiple models with similar data sources, that is to say the same structure and the same information background. And so this allows to reduce training time and costs. And eventually we can also improve the quality of the data by providing some feedback with rules and constraints. So that's the big picture. But how to collect, manage, and provide fair data? In the context of open sciences, making data fair is about data being as open as possible and as close as necessary. This is for openness. But fair and open are not synonymous, as data can be both one or neither even. So, however, the FAIR principles can be applied to digital resources to make them fair by design, in order to improve reproducibility and transparency as well. So in the context of data-driven research and open sciences, we need to formalize knowledge to make it explicit and shareable. That means not just the data, but also information about data, that is metadata, and this is to share the context and provenance of the data once again. So for this, ontologies are a key solution because they are providing interoperability and reusability. And why are ontologies so helpful? It is because on the lower level here, you see the syntactic interoperability, which is providing a consistent data format that can be interpreted by different systems. But on the highest level, we have the semantic, we have the semantic interoperability, and this is leaving a few room for error or misinterpretation, because semantic interoperable systems share the same conceptual understanding of what the underlying data conveys, thanks to ontologies. Ontologies, they are the most refined semantic artifact, as they have some rules and constraints, and they also have some specialized relationships between entities. So however, there is a broad spectrum of ontologies, and you may distinguish between upper level, that is foundational ontologies, which are very formal standard. And then we have mid-level ontologies, that is to say core ontologies or task ontologies. They are dedicated to some purposes. For example, you will see that we are reusing SOSA-SSN, but also time ontology or provenance ontology, which are different kind of mid-level ontologies. And finally, the application level, we have the domain ontologies or terminal ontologies, which are providing specific vocabulary or vocabulary specific to the field, providing labels and terms and definition for human, at least, understanding, and also for machine. They are also machine actionable. So it is to say that we have two domains ontology here, PO2 TransformON and FermentON, which are both based on the PO2 core model. And so ontologies allow us to reach semantic interoperability. So now I will show you the process and observation ontology. This is a very simplified view. The ontology is a generic model in all, the semantic web standard for ontology. And we are reusing SOSA-SSN concept here in green. It is symbolized here in green. And we are also reusing other ontologies, such as BFO, IAO, time ontology, QDT for units, and metadata schemas, that's like DCAT or schema.org, and the unified code for units of measure to standardize the units. And this generic model was designed to describe processes as sequences of steps here in blue, and input and output components. And this is the special PO2 core concepts, and then we are reusing SOSA-SSN concept for observation with materials and method. And the results finally are described in data tables at the instance level. And the quantitative and qualitative values in the data tables are conforming to the I-adopt variable design standard patterns, which are offering some patterns, some standards for representing the data. So PO2 provides a generic pattern for developing domain ontologies and structuring the data in the form of annotated data sets, which are in the RDF all formats. So now I show you the upper level hierarchies of the domain ontology TransformON, which is specializing the core, the PO2 core concepts. So it is a unified vocabulary that provides all the concepts useful to describe transformation process and also characterization of food, feed, bioproducts, and biowaste. And as you can see, it is organized in three main parts. You will recognize the blue part I showed you before in the schema. And this is the process part. The result part, which assemble all the description, the descriptors or attributes we can apply on the data. And finally, the green part is the observation part. And as you can see, we have the main core concept here, which are specialized. And we have PO2 component, PO2 process, PO2 step, the PO2 attributes, and finally in the observation part, PO2 material, method, and PO2 scales. And finally, steps are the elementary operating condition. Here you are only seeing the upper level, but we have a deep hierarchy behind. And you will see also that we can describe both measurement instrument or processing equipment as well as different scales. And this is the originality also of the model with respect to SOSA-SSN, because this is an originality as far as we can describe both measurement scales or process scale of the process. And a variety of use cases and research objectives have already been addressed. So I will show you just briefly some example here, but you will find more detail in the publication which are listed on the slide. So briefly, for the first example, the objective was to predict the performance of the microfiltration process. And here the problem to be solved is that we have very heterogeneous and incomplete data, and existing models are limited to a specific range of operating conditions. So we have to assemble many data sets to have sufficient data to make modeling. The second example is to optimize composite food packaging formulation. And here the problem is that we have to find what biomass and what loading rates to achieve for some desired mechanical characteristics of the end product, which is the composite food packaging. And as you can see, we have to make a decision about how many PHA or PHPV biopolymer we have to add to obtain this desired mechanical characteristics with respect to the biomass, which we can add as filling material. And finally, then the third example now is to study the environmental impacts of cheeses. This is to speak about interoperability between two information systems, namely our knowledge bases is providing the process flow charts and the life cycle inventories data needed for the life cycle impact assessment, which is made by using the means. This is another information systems tool, the means in-out software, which is Mainly, briefly, it is for giving access to the CIMAPRO LCA software, and then give LCA data back. So this is also described in this publication. And finally, if we want to design healthy, sustainable, and acceptable food, we have to relate transformation process and some nutritional or food composition data and sensory data as well. So the problem here to be solved is the integration of heterogeneous data to provide a global quality score. So the main added value is to be able to reassemble data, knowing the conditions under which they were obtained. And as this data have been structured with the PO2 model, this data can then be mobilized to generate knowledge about complex systems. And using advanced data processing techniques, such as Bayesian networks or other machine learning, we can improve knowledge. So now, what's next? I hope it is not too quick. I have to speed up, correct? It is now to see what is the ongoing project FermentON, which is also a project funded by INRAE Research Division, MICA, and Transform. So in FermentON, we want to extend the domain ontology PO2 TransformON to include processes involving microbial agents and also enzymatic reactions occurring during biotechnological processes. So we have to describe fermentation processes and also more broadly biological processes. And as you know, we have the biomass as inputs, then we have the process key parameters, namely the operating modes and downstream and upstream treatment as well, to have the end products, and we want functional end products. So we have some targeted characteristics as outputs. And here, what is novel is that we have to deal with the degree of complexity and diversity of microbial community, and especially regarding their metabolic pathways. So now some example of question to be answered could be, what combination of substrate, agent, target product are we want to make regarding the available equipment, and would, for example, for a specific step, that is to say, the cultivation method. So we have to know which are the relation between these entities to be able to decide. And for example, another example would be what products result from metabolic activity, or intermediates or metabolites are produced, and which with which enzyme. So I think these are the kind of question you are interested in. And now we have some use cases also, of course, and the general aim of the project was to be able to combine different types of data, both on process data and characterization data, and both for food and non-food applications. So we have first the food fermentation use case. So we have some products as for example, cheese, mixed dairy and plant-based yogurts, or fermented vegetables. And we have also the winemaking, which this is for fermented food and beverage. And for winemaking, we have a continuous line from vine to wine, and a multi-scale study of alcoholic fermentation. And then for the bioproducts, matter and energy side, we have the composite making, which is the production of PHA, PHBB for the composite packaging materials. And for environmental biorefinery and biotech, we have a specific use case on electrofermentation, which is a quite innovative process, I guess, and also the design of soft sensors. And Camilo, I think you are here. So now the scope to be covered in these projects, in this specific domain ontology, we will extend the vocabulary of TransformON to the concepts dealing with microbial agents, namely the degree of complexity of the microbial community and role, so the role of microbial agents in the process, because the core scopes is the process key parameters, as well as the different types of substrates. And depending on the complexity of the biomass, we can have several degradation or hydrolysis stages and intermediary products, and then several, of course, several microbial species involved. And finally, as omics data are not within the scope directly, as these data are already managed within the bioinformatic communities and are deposited on specific data warehouse. But however, we will deal with the species or strain of microbial agents, let's say by using meta-barcoding results, and also with the phenotypes. So actually, the aim of the project is also to be able to enable interoperability between different information systems for data retrieval of microbial phenotypes and targeted molecules or compounds. And so we will be very focused on the description of the environmental factors and process key parameters, for example, the description of the bioreactor and associated sensors. So that was quick, but now I have to leave the floor to Julien, which will he will present you how to produce fair by design and AI-ready data in practice. So Julien, I think you are ready.
Yes.
And stop my sharing.
I will try to share my screen. Up. Okay. And now I think you can see my screen.
Yes.
So thank you, Magalie. Now we talk about how to produce fair by design and AI-ready data in practice, and we will dive into the software ecosystem. First, I want to come back on this schema. Magalie show you the simplified ontology representation of the model. You can see this on the left in this slide. It's a simplified representation because there are a few concepts and relations, but the reality is far more complex. As you can see on the right of this slide, there is a lot of concept and relations. And to handle this complexity, we need the information system, and especially ontology-driven information system. So we need this kind of information system for producing fair data. And this information system addresses main needs like data entry, data querying, and relies on a software architecture that will ensure security and access management, and also interoperability. So the first need is the data entry. For this need, we develop a desktop application called PO2 Manager. PO2 Manager have some main feature. Through this application, you can entry data, and you have also visualization of the process directly through the application. I will show this in the next slide. You can also import and export Excel template files to fill the model directly through the application. We have data quality checks, so terminological consistency checks, and also data checks. For instance, we can check if a value is in a specific range or things like that. And we have also a semantic process. It's important to note that PO2 Manager is a desktop application, but all the data and ontology are saved on a remote server, and you can use the application to save and share data directly to the application. The benefits of using this application, it's ontology are stored on a centralized database. This is also an important thing because this choice make the data consistent across different sites and contexts. So when we add something in the ontology, we add it for all the context and all the users. So you don't have a duplicate concept or duplicate relation between the different installation of the PO2 manager. We have a good data understanding because there is some visualization feature to see the studied process. The vocabulary is standardized, but there is no semantic prerequisites required to use the application. The vocabulary is accessible directly through the application. And it's also linked with international W3C standards like semantic sensor network ontology and time ontology. So now I will show you some capture of the application. So this is a capture of the PO2 manager user interface. And here, this is a capture where you can see a process itinerary as a graph. So you can see in black some steps of the process, in red the components, and in black and green observation. So you can click on the different elements on the graph and see more details. This is what we do here. Here, for instance, we have a zoom on the specific step. And the important thing here is through the form of the application, there is a mapping between the field of the application and the underlying ontology. So here, if you fill material and methods, you are feeding the directly data with structure with the SOSA ontology. And here you can see the specific step, material method involved in this step, and also input and output components of this step. The next slide is quite the same, but here it's another zoom on the observation associated to these steps. So you can see here observation, PO2 observation, and the link between the observation and the underlying ontology SOSA. I want to mention that this application is in production and has been already used to produce dataset and to upload this dataset into the INRAE repository called Recherche Data Group. For another need, we have the data query need. We have developed another web application called Spock. The Spock application is a web application and he has main feature like the search engine with two modes. A simplified mode for standard users, where you can use the different web forms to request the data. There is no technical knowledge required to use this application. And we have also an advanced mode for power users. I'll show you this on the next slide. And in this mode, you have you can write a complex Sparkle query and perform some fine tuning of the query. The application provides also automatic conversion, so you can query data with different unit of measure. You can save the most frequent queries for execution, and you can also export the query result into different formats, CSV, JSON, and so on. So Magalie told me that you talk about raw crate format yesterday, so I think it will be we can easily generate this kind of format because everything is based on semantic standards here. So the benefits of using this web application, it's we are using controlled vocabulary to query data. We can query public and private projects data. I will zoom on this point in my next slide. And you can perform query between different open data sources. So there is a capture of the POC user interface here. It's in simple mode, so you can see that you have to select dataset, you can select a data set and also define some filter on a specific set, and also filter on the observation. So you can here restrict the set of observation associated to a specific criteria of volume here. And then you can execute the query through the button on the execute form and retrieve the data in CSV or JSON format. So here it's a capture of the advanced mode. It's important to say that Sparkle is a powerful language to request data, and sometimes for power users, it's frustrating to use the web forms to build a query. So here in this mode, you can write query not buildable directly by the form, and you can perform some fine tuning. And this mode unleashes the full potential of Sparkle, so you can perform some federated queries across different data sources and obtain the query result in the same way than in the simple mode. So you can save the result in CSV files or in JSON LD. This is quite barbaric, but it can be easily managed by advanced users. Here it's a slide about the security and access management. It's important to note that the data is fair, but it's not necessarily immediately open. So you have the possibility to create a private project and to host your own private project datasets. The account creation is also flexible in the sense that it's not necessarily to be affiliated to an institute to use the ecosystem. So we allow private partners to create accounts. And for the administrator of the dataset, you can manage read and write permissions. And you can also query private and public data at the same time. And just a word about the technology used. We are using common open source components. The software is written in Java language, and we are using GraphDB for the triple store. It's the database in the semantic world. And everything is deployed through Docker, so it can be easily deployed. We can easily deploy a new instance of our ecosystem. And the last point is the what's next. So we are working on a workflow in order to integrate contribution and improve the domain ontology by integrating a scientific point of views. And the main idea here is if something is missing or incorrect in the ontology, you can suggest a fix or an add by creating an issue on Git and GitLab. And we want also to develop and reinforce AI applications. Today, we are working on a workflow to generate constraints, combining PO2 ontology and probabilistic relational model. So we generate some constraint on data through Shackle constraints. And we want to use it more massively. And we are also working on a large dataset management. So we want to be able to perform some semantic queries on a large dataset, and for instance, on long time series. And we are working on a proof of concept. I call this best of two worlds, because the main idea is to reuse the big data technology stack for performance and to use semantic technology stack for knowledge representation. So there is a framework called Sensa who offer this kind of functionalities, and we want to use it to try to request some large data set with semantic technologies. And the last point is an interface improvement. So we want to include some UX design to improve the usability of the web application, especially because it's the newest application. So I can let you conclude, Magalie, if you want. I will stop to share my screen.
Thank you. So I don't see you, sorry, but yeah, now. So now in a nutshell, so to conclude, to summarize our workflow, we have structure of data of interest thanks to the PO2 core model and domain ontologies. Then we have standardized metadata thanks to the domain ontology. And we have the possibility to query and link dataset thanks to the W3C standards. And so we have our PO2 core and domain ontologies, which are all standards, W3C standards. And we have the PO2 manager tool, the first tool to structure the data and annotate the data with the control vocabulary. And then we have the second tool, the Spoke tool, which is enable us to query the data which are stored as semantic resources in RDF format in the GraphDB databases. So we have some AI-ready data we can share and reuse. And this allows us to make some knowledge discovery thanks to statistical modeling and machine learning, and which were not presented today, but which are able to, which you may know. And finally, we have the open data publication option, because we have fair by design data which can be deposited finally in the repository. So for example, the data INRAE is the Dataverse collection in the research data group, which is a French data repository, and also known as the Dataverse project. And so to answer some questions in the chat, the PO2 Manager and Spoke softwares are available on Software Heritage and the INRAE repository. INRAE is a hypertext archive. This is the institutional repository here in France, but it is open. And all the details of the ontology building work and the data sets from the use cases I show you are detailed in this paper. And please contact anyone of the contributors listed in this paper if you wish more information after this talk. And we are also proud to say that this work was rewarded in 2023, so last year, by the French Minister of Higher Education, Research and Innovation. And this is in the framework of the France second plan for open science and the INRAE funded data source food project. So in conclusion, the PO2 transform ontology has been designed as a solution for integrating different information systems of knowledge bases. And it provides the vocabulary needed by the community, validated by a collective of domain experts. And the vocabulary and the ontology is flexible and well adapted to different granularity levels required in different research projects. And this is, in the meantime, we are providing a flexible but harmonized structure. And we have also the background information needed for further processing. So now I am ending this talk by presenting you all the team. And this is the core PO2 core team, which includes experts in knowledge engineering and computing from Transform and also the EMIA Paris-Saclay unit. And this is the members of the CATI DISCO I told you in the beginning. And so you will remind us, and please documentation and support are here. So don't hesitate. I will leave the slide at the end, and you will have some links here to have the documentation. And so happy to answer to question if any. Thank you.